Hi students, welcome back. Here I am, your own biology teacher, Dr. Mahmood Hassan, with another informative and easy to understand video. In the previous video, we discussed nucleic acids, their types, and chemical structure of molecules that form nucleic acids. We studied how purines and pyrimidines are numbered to fully understand the difference in the chemical structure of 5 nitrogenous bases so that we are able to draw them confidently on the paper without any mistake. Today, we will study how these biomolecules combine to form repetitive units of nucleic acids called nucleotides. Before we move forward, I once again remind you to subscribe to my channel Biology for All and like this video. Please watch this video till end so that we can understand their structure and get good grades in the examination. Oh, by the way, do you see the two men in the picture? Do you have any idea who they are? These are Mr. Watson and Mr. Crick in 1953 in their lab with the model of the DNA that they presented to the public. Let us start with our topic. We have two types of nitrogenous bases called purines and pyrimidines, two types of pentose sugar, the ribose and deoxyribose, and a phosphate group that chemically combine to form nucleotide. Purines have two ring structure, one hexagonal ring containing 2 nitrogen atoms and 4 carbon atoms, and another pentagonal ring containing 2 nitrogen and 3 carbon atoms. Purines are found both in DNA and RNA. There are two types of purines in nucleic acids. These are adenine and guanine. Numbering in purine starts from nitrogen at ortho position. Technically, adenine is 6 amino purine, while guanine is 2 amino 6 oxypurine. This means that adenine has an amino group at carbon number 6 and guanine has an amino group at carbon number 2 and an oxygen at carbon 6. These differences are shown by the blinking triangles in the slide. Pyrimidines have a single hexagonal ring with 2 nitrogen atoms and 4 carbon atoms. There are 3 types of purines in nucleic acids. These are thymine, cytosine, and uracil. Thymine and cytosine are found in DNA, while RNA contains cytosine and uracil. Thymine is absent in RNA, while uracil is absent in DNA. When these nitrogenous bases chemically combine with sugars, they form new compounds. In DNA, adenine becomes deoxyadenosine, while guanine becomes deoxyguanosine. Similarly, names of purines also change. Cytosine and thymine become deoxycytidine and deoxythymidine. In RNA, however, there is no deoxyribose, so adenine becomes adenosine, guanine becomes guanosine, cytosine becomes cytidine, and uracil becomes uridine after combining with ribose sugar. Now let us see how these molecules combine with each other. When a purine combines with a pentose sugar, chemical reaction occurs between nitrogen at position 9 of a purine and carbon 1 of a pentose sugar. Hydrogen atoms attached to nitrogen 9 of the purine combines with hydroxyl of carbon 1 of pentose sugar. You can see in this slide when an adenine combines with a deoxyribose, a water molecule is released giving rise to deoxyadenosine. Guanine also follows the same pattern to form deoxyguanosine. When a pyrimidine combines with a pentose sugar, chemical reaction occurs at its nitrogen 1 and carbon 1 of pentose sugar. Hydrogen atom attached to nitrogen 1 of the pyrimidine combines with hydroxyl ion of carbon 1 of pentose sugar. You can see in this slide when a thymine combines with deoxyribose, a water molecule is released, giving rise to deoxythymidine. Cytosine and uracil also follow the same pattern and form deoxycytidine and uridine. The last step is the binding of phosphate group at carbon 5 of pentose sugar. With the completion of this step, nitrogenous base becomes a nucleotide. Thus, adenine, guanine, cytosine, and thymine in DNA become D-adenosine monophosphate or DAMP, D-guanosine monophosphate or DGMP, D-cytidine monophosphate or DCMP, and D-thymidine monophosphate or DTMP. While in RNA they are called adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate, and abbreviated as AMP, GMP, CMP, and UMP respectively. This ends up today's lecture. Please provide me your feedback through your comments and suggestions, and don't forget to subscribe to my channel Biology for All. Bye for now. See ya.